The overall goal of this procedure is to characterize the distinctive biology at the interface between malignant epithelial cells at the invasive tumor front and cancer-associated stromal cells. This is accomplished by first establishing primary fibroblast cultures from colonic explants. The second step is to construct organotypic co-culture models using primary colonic fibroblasts and cultured colorectal cancer cells. Next, organotypic models are fixed in formalin, sectioned onto membrane-mounted slides, and stained with crescel violet. The final step is laser microdissection in order to separate invading tumor cells from non-invading tumor cells and cancer-associated stromal cells. Ultimately, molecular profiling techniques may be used to characterize the biology of invading and non-invading tumor cells and stromal cells in the surrounding tumor microenvironment.
We first had the idea for this method when we realised how clearly distinguishable invading cells are from non-invading cells in three-dimensional organotypic co-culture models of colorectal cancer.
To begin, obtain samples of normal human colon mucosa tissue directly from the surgical operating theatre. In the laboratory, place the specimen at the center of a 10 cm tissue culture dish. Then wash the culture 3 times with phosphate buffered saline, or PBS, supplemented with penicillin, streptomycin, and fungazone.